All right, and now for something completely different. I say that because unlike the last speaker, not only do I have little to say about morality, I think it's pretty fair to say that I'm probably one of the more amoral individuals you will ever meet. And so in this regard, you know, it actually provides a nice contrast, because on the one hand, liberal agenda or not, there is a certain humanist body of thought, and that is interesting to examine. But I think I come from another strain, as it were, of non-religion. I'm more a technocrat. For me, I'm going to show you your future dystopian or not. And you get to decide, fortunately, because in a rational republic, still the citizenry decides what people like me do. But nonetheless, I'm just more along the lines of, here's what we can make, and I'm just going to give you an update. This is also along the lines of scientists are really, really lazy, because all I did was slap together some of my slides, and so this will wash over you like a giant sea of information, some of which is comprehensible and some of which is not, and you shouldn't really worry about that, because any good lecture really is performance art. You can judge me on my performance art, but you can just decide whether this vast sea of information just rolling over you is pleasing or displeasing or whatnot, and ask me questions about it at the end. In this regard, though, I'm going to talk about evolution in the test tube and sort of in the wild. By in the wild, it's not going to be all that wild. It's going to be, you know, wild bacteria. But nonetheless, you know, woo! But nonetheless, I want to give you an example of how things we do in the lab can translate into living organisms. All right, now first what I'm going to do is talk about something called synthetic biology. Now this is a buzzword that's been buzzing about, and what it really is the reincarnation of what we used to call bioengineering, and before that molecular biology, and so on and so on. And there's nothing new under the sun. We've known for a long time that we can engineer organisms. The Victorians knew it quite well. They made many, many, many breeds of dogs and horses just by simple breeding experiments, and that's as much a man-made innovation as anything I'm going to talk about here. But what is available to us today is the ability to make things almost from scratch, to make DNA, to implant it in organisms, and to change them thereby. And the engineers have finally cottoned to this and are building from scratch various degrees of complexity. So this is sort of an engineer's fantasy of what biology should be like. We start down here with very small things, and we engineer all the way up to potentially ecosystems. Now, as a sometime evolutionary biologist, I don't think it's going to work precisely like this, but in fact, there's more truth to this than not, that we can build from scratch systems and then use evolution not as a natural process, but as a tool to refine them. So I'm going to talk about engineering feats with evolution. And first I'm going to go from large to small, and then I'm going to go from small to large. There's going to be two stories here. Both of them you should note equally incomprehensible. All right. And in going from large to small, it is fortunate that we had some introduction to the bees in the last talk, because we're going to talk about bee guts just a little bit, and then we're going to talk about addiction in bee guts. All right, now this is a project I'm carrying out with several other researchers at the University of Texas at Austin. It's called Control of Bee Behavior by Stably Engineered Gut Microbial Communities. And again, we heard in the last talk that to some extent, our behaviors, our phenotypes are regulated by our commensals, by the organisms that live with us. So there's bunches of organisms on our skin, in our guts, everywhere throughout us. In fact, there's actually organisms that probably, for example, if you were a cat, there's a particular organism that lives in a cat's bloodstream. That's why cats can give you cat scratch fever, is because they have a particular organism that actually lives with them. Cats, it's sort of like cat's own form of biological warfare. And so we hope to engineer these commensals, these microbes, to make bees do interesting things. Purely for the sake of making bees do interesting things, refer to previously said statement about me being a technocrat. All right, here's the team, not all of whom by any stretch of the imagination are tech. Oh, wow, what did I do? I destroyed everything. So we don't, okay. Don't care. All right, so I'll just point with my hand. There's Nancy Moran. Nancy is awesome. Nancy is the bee person. She understands a huge amount about the bee gut and about what happens in bees. There's me. That's Hal Alper. He's actually a real true engineer. Jeff Barrick, a real true evolutionary biologist, and Brian Davies, a real true microbiologist. So this is our team to help understand and engineer the bee gut. Now, this is a slide I put in this morning, and this is actually a slide that sort of generically represents so-called control theory. And if you're an engineer, you know what control theory is, and if you aren't, you don't, and you probably never will. But what it really means is that there's a theory for, wait for it, how to control things. And what this theory says in part is you have a controller that is controlling a process that has some way to sense what is going on. Filters may or may not be necessary, and then you feed back on the control. All that really says is that's the most schematic diagram you can possibly imagine for how you might want to control something. Now, as organisms, we have lots and lots of this inside us, but of course it's all integrated, and it's all so far been subject to natural selection. It's adapted for whatever we've adapted for. As an engineer, or as someone who likes to tinker with things, we'd like to put in our own controllers, right? And to do that, we have to somehow have a orthogonal or alternative control system. So this is our great plan for having an alternative control system in the bee. What we'll do is we'll take the current honeybee gut microbiota, the bugs that live in the bee, and we'll get them to be codependent on one another in ways we can control. We'll make engineered consortia, and I'll tell you a little about the engineered consortia in just a minute. And the engineered consortia will live in the bees and potentially control the bees' behavior by making peptides or neurochemicals that will cause the bees to do different things. So in a reverse of how we normally think of the great scheme of being, the bacteria will control the bees, and the bees will control the hive. So we're going to go from the molecule to the cell to the organism, and the organism to the social organization. Now, we haven't done this yet. This is just our plan. And again, we're engineers. We like working down here at the molecule level, but we have big plans for how we're going to get all the way up to the hive level. It should be pointed out, though, that this is just a great system to work on because there really are just 8 species. In us, there's hundreds, thousands of different species in our gut, but in bees, there are really just 8 generally species, and we know exactly where they fall, and we know a lot about them. We even know several of their genomes at this point, and they live here in the bee's hind gut, which, you know, to a first approximation as a technologist is, that's a bee's stomach, sure. I'm not a bee person. What we want to do is make this control vector, this alternative control system. We call it an archmid because it's going to carry lots of things into the bee. It's, you know, 2 by 2 will go our genes into the bee, and on the archmid will be all the control structures. But the idea, and nicely represented even by these simple schematic arrows, is the vector will control itself, and it will be independent from the bee's control systems. It will be a superimposed and orthogonal control system that will act on the host. Here's a more complex diagram of the archmid, and again, you don't need to know what any of these arrows mean. Heck, I don't even know what most of these arrows mean because I make them up as I go along. That's not entirely true, but that's the great thing about being a technologist. Rather than do dissective science, oh, you know, nature and or super being gave me B, now I can study B. Boring. Why don't I make something new, right? And it's like, well, maybe the reason you don't make something new is because bees are pretty cool. And so sometimes the way I say it is, I would rather engineer a sundial than take apart a watch. And so blind watchmaker or not, there's an advantage to being a sundial maker. You get to eventually make things more complex than sundials, and you get to control them. So in any event, this is going to be our more complex archmid. One of my roles in this is to make the engine of the archmid, the engine that will drive gene expression only for the archmid, separate from the bacteria that lives in the bees, and certainly separate from the bee itself. Jeff Barrick's role in the project is to slow evolution down, which is counterintuitive because normally we just think of evolution as something that happens. It goes at a particular rate, there's selection, you have fast bunnies, you have slow bunnies, you have polar bears, whatever. But in fact, it's something that is subject to mutation rates, to selection, to how you interact with your environment. And so what Jeff is trying to do is slow the mutation rate in a given organism. And there are organisms with faster mutation rates and slower mutation rates. It is subject to engineering. And so Jeff has been, for example, selecting for organisms that make a particular amount of green. It really is like that. It's about that simple. And if he selects for something that makes a particular amount of green over a very long period of time, then what he's doing is essentially establishing an evolutionarily stable state. And we're trying to figure out what genes have changed to establish that evolutionarily stable state so that we can force, when we put these things in the bee gut, our archmid to be stable. Because the bee gut microorganism may say of our archmid, oh, I don't really like you. And we will say, we really don't care because you can't evolve anymore. And so we're trying to make these evolutionarily constrained vehicles almost counterintuitive to the theme of Darwin Day in order to better engineer the bee gut microbiomes. Now this is truly brilliant. This is the innovation of Brian Davies, the microbiologist. And one question is, we're going to put these 8 microorganisms in. They're going to have their own ArcMid, their own alternative control system, about which I'll tell you a little more in just a minute. And they're going to have a slowed evolution so that they don't kick it out too readily. But what if 7 of the organisms don't like the other one? Or what if one of them decides to go rogue and take over? How do you keep a consortium of cells interdependent on one another? Now, they learned through evolutionary time to be codependent within the bee gut, but now we're taking the fetters off. We're making things that can do all sorts of new and interesting things in the bee gut. How do we know that consortium can be enforced? And so again, as engineers, we're excited about the notion that we can force interdependence, not just wait for it to evolve over long periods of time. And Brian had this particularly nice idea for forcing interdependence. Basically, we'll get each organism to hold a gun to its head, and then we'll have its friend talk it down from the ledge. And we'll do that in a circle. So organism 1, ready to commit suicide, will be talking to organism 2 about not committing suicide. Organism 2 will be talking to organism 3 about not committing suicide, who will talk back to organism 1. In a round chain of events, all the cells will prevent one another from killing each other. And because of that, they will, in fact, be interdependent. The way they're going to kill each other is Brian has actually figured out how to put toxic proteins up on the cell, on the surface of the cell, so they slowly kill the cell. Not immediately, but they slowly kill the cell. White knight from adjacent, a friendly cell, makes a protease that can cleave, can chew up the death peptide. So again, it literally is, I have a gun to my head, I have something that is killing me, and here you are talking me down, eating the peptide that would otherwise kill me. So Brian's in the process of implementing this, and it's going pretty well. He actually has the suicide part of it down pat. And so he's figured out how to get cells to kill themselves, albeit somewhat slowly. Now, again, in dealing in general with amoral individuals and technologists, there's a certain intake of breath where it's like, oh my gosh, how can we most efficiently put regulations in place of this person? And in fact, I assure you, I have many regulations in my way. But the advantage, I horrifyingly have to tell you, the advantage of having, dare we say, free thinkers of whatever stripe is that oftentimes those free thoughts lead in all sorts of directions, not just amoral ones. And so what is the other advantage, potentially, of a suicide peptide that allows you to enforce a consortium of bee gut microorganisms to control the behavior of honeybees? Oh, wait, you just said it committed suicide. Maybe it can commit murder, too. Yes, it can. So Brian has a generalized method, not just to find peptides that kill organisms that have that peptide, sort of useless unless you're in a consortium, but to kill other organisms. And so Brian's developed a whole new class of antimicrobials that actually have very broad spectrum and that can be used for controlling nosocomial infections in hospitals, for example, or to take out particular flesh-eating bacteria that may be, say, eating your flesh. And so in this regard, you never know really where inquiry is going to go. And again, I fall back on my technologist roots. All right, so you get the general scheme, I hope. Things live in the gut. We're going to give them an orthogonal control system. We're going to teach them to be interdependent, and then they're going to help control the bee, which will control the hive. I haven't talked so much about the control upwards because we're still establishing the internal control, the ArcMid, the self-suicide and assisted help system, and so forth. But my contribution to it, again, lazy scientist showing you his own slides, is to help addict the control system to the bacteria so it can't get rid of it. Jeff Barrick's going to slow down evolution so that my addiction doesn't have to be perfect, but I'm trying to addict things in there so that they use what I want them to use. And so I'm going to digress a little. While you may not learn much from this talk other than, you'll never get this hour of your day back, I'll throw in a little knowledge here and there, right? As the great sea of information washes over you, every now and again there'll be knowledge. And so one piece of knowledge is, this is the genetic code. The genetic code is so cool. I cannot tell you how cool the genetic code is. Because out of these 4 letters in combinations of 3 to make 64, 4 to the third, you get translation into 20 amino acids. And this is just sort of a pseudochemical representation of those amino acids. But what we're really saying is you got a program for Legos. right? Here is the code, there is the Lego, and so what we're going to do with that code is we're going to put it together. And that's what translation is. All of your proteins, which make up most of what you are, is encoded in your DNA via the genetic code. And that genetic code is read out by a beast called the ribosome, and that beast makes proteins one at a time, from one amino acid to one amino acid to one amino acid. It reads the code, reads the code, reads the code, spits out a long protein. But it can only do what's in the code. If you want to make this is the code for this, and you put it in at the locale that it should go in. But what if you want to change the code? What if you want to make something new, different, fun, fast? You have to change the code. You have to engineer translation. And all that really means is you have to engineer the machinery of translation to put in something new. And so that's where we come in. My lab has been working on engineering the genetic code to take over some of the less used portions of the code. It's as though you're coding actually I've been trying to code in Python recently, which is a little like monkeys typing Shakespeare. But nonetheless, I come across these commands in Python that I don't use very often, right? They're probably useful for some programmer, but I don't use them very often. And I sometimes think, well, what can I do with that instead of just what Python wants it to do? And similarly, if you have the genetic code, there are these stop signs that don't get used very often. They get used some, but not all that often. And so we're going to take one of them over, and the stop sign will now become a new amino acid, a new chemical, a new Lego block in the cell. And so, in fact, we're going to encode for nitrotyrosine. And nitrotyrosine here is an unnatural amino acid. And what we did was we said, well, now we want to addict the organism to nitrotyrosine so as to better establish control over that organism in the bee gut, in the microbiota, in the bee, in the hive, right? From the bottom all the way to the top, we're establishing control, and we're going to establish chemical control by addicting it to this chemical. And the way we addict it to this chemical, directed evolution comes in here, is we're going to put it in a pocket, and then we're going to evolve the pocket around it, right? So nature said, look, over several billion years, I have a perfectly decent pocket here. Would you please not mess it up? And we're like, no, we're going to mess it up. That's too bad. And so we put it in there, and it's like, you've messed me up. And we say, that's okay. You get to fix it. And we throw fast forward on evolution. We do directed evolution procedures to relax the pocket. And what Drew Tack in my lab did is we came up all these different solutions. These are just the code read out in things that have fixed the insertion of the unnatural amino acid. And he looked for particularly good fixes where it looked like it was only fixing around this new code. And then he proved it by saying, here is the fix. I've put in this. And it could revert to any of these other things, right? It could mutate back, although we hope Jeff Barrick makes it really slow evolution. But you can see if this is what you need to live, then there's lots of stuff that makes you die. There's one other good thing that helps you live, but in fact, it's 2 mutations away from this. You have to have 2 simultaneous mutations to get from here to here. And these other things, it just doesn't work very well. And so what that means is it already is slow evolving because it's really hard to pick up 2 mutations at the same time. And all these are growth curves that over multiple generations, actually out to 250 generations, this thing does not revert. We have addicted the organism to a new chemical. And so therefore, if we put our archmids carrying this addiction into a bacteria, the bacteria needs our chemical, and it needs our archmid, and it needs everything that we give it, and will stay in place, and Jeff Barrick will slow evolution down, and this will be the house that Jack built. One of the great things about this is, remember, I talked about an orthogonal control system. That means we should be able to move it around. Well, having one protein that's very, very important addicted to this chemical means we can move the chemical from one strain to another. And so Drew actually moved from E. coli, a strain we typically use, to a variety of organisms, including bee gut microbiota, bee gut organisms, to show that we could move the addiction. This thing is essentially crack for bacteria. You use it once, you're on it forever. Okay, and this just says what I already said, that we're going to move this into the bee gut. We're also moving it into the mouse gut to see if we can have addicted organisms in the mouse gut. And again, this is one of our schemes for establishing orthogonal control. Part of that was engineering, synthetic biology, where we say we can recreate life as we wish. And part of that was we needed directed evolution to do it. We couldn't just design it. We have great plans, and we are good engineers, but we still need to use evolution to get and make some of these things. All right, so that was from large to small. Now let's go from small to large. And so we're going to talk now about fundamentally changing the tree of life, changing the code, again, just like we did for the bees, and how that's going to play out not in bees and bee guts and mice and whatnot, but in bacteriophage, which are the viruses of bacteria. So it's really tiny, and it's sort of like, you know, is that really life? And it's like it's fun, so just bear with me. Now, I got this off of Facebook, which I'm not on, thankfully, this morning. But my wife is on Facebook, and you know, she said, "Oh, look, Dave Hillis posted something really cool this morning for Darwin Day," and he did. And so I thought I'd steal it. So this is the poem "The Tree," as adapted by David Hillis, with apologies to Joyce Kilmer. I think I shall never see a poem as lovely as the tree of life, the tree that relates all of life with likelihood or Occam's knife. And there he's alluding to the fact that sometimes it's difficult to tell exactly where to put something in the tree. The tree that shows us of archaea, fungi, frogs, and protostomia. The tree that grows through all of time, relating all your groups to mine. Now actually this was really small print. It's a little bigger here for you. And I originally thought this says the tree that grows through all of time, making all your groups be mine. And so for those of you who are video game players, you may remember, you know, all your bases are mine. And so this, I thought that was what Dave was riffing off of, but he wasn't. Relating all your groups to mine. I think mine's better. Upon whose branches new clades are born, extinction too where lives are shorn. Because of course the tree of life is not a tree of forever. Things die. New things evolve. Poems are made by fools like me, but only Paup can make the tree. This is homage to somebody who actually develops phylogenetic methods for computing phylogenies. So I just thought that was a really cool poem for Darwin's Day, but it also is a lead into the tree of life, of course, relates us all. Underlying the tree of life at some level is the code of molecular biology. And I've already alluded to that code in the sense of the genetic code, but there's an even more fundamental sort of mechanistic code, which is called Crick's central dogma. And in Crick's central dogma, DNA makes DNA, makes sense. DNA makes RNA, okay, I'll go with that. And RNA makes proteins here. But basically, there are individual machines, and if you want to change the underlying machinery that is also underlying those trees, you have to work at it. And so one of the questions my lab recently asked is, how do we turn something that makes DNA from DNA into something that makes RNA from DNA, a so-called reverse transcriptase? And so I thought since on my Mac this looks good and on PCs this just looks horrible, I apologize. But basically in the tree of enzymes, in the tree of machines that make things, there are DNA polymerases and there are RNA polymerases and there are so-called reverse transcriptases. Now reverse transcriptases make DNA from RNA, and they're mainly found in viruses. And they're mainly found in viruses because viruses have weird life cycles where they'll be, I'm coming in as DNA, then I'll make some RNA, then I'll make some DNA. Whereas we, and literally every other organism, like I'll make DNA from DNA from DNA from DNA, I'll make some RNA, that'll go make a protein. I'm never going to put my precious genetic information in RNA to be copied back into DNA. That would just be dumb. Why are you doing that, virus? But we decided to see if it really was so dumb. And we decided to make a reverse transcriptase from a DNA polymerase. So the machines that normally make DNA, we're going to try and get them to pretend to make DNA from RNA. And to do this, to change the fundamental biochemistry of a system, to change the tree of life, we had to evolve dead things. And that just means that nature moves slowly. I didn't really talk much about directed evolution. I said we could direct the evolution of translation to make unnatural amino acids part of organisms in the bee gut. But I didn't really tell you much about how we did it. Now I'm going to tell you a little more about how we do it. And what it does is it involves saying, if you take the rules off and you're going to evolve underlying biochemistry or mess with underlying biochemistry, you can't be waiting for things to live. And so this is our scheme for evolution. And what we do is we make a bunch of different variants of a machine. That's really all it is. We've got a bunch of machines, and each machine is in a different cell. That's supposed to be what's represented there. And you can tell by the colors that the machines are different. You got your green machines, you got your red machines, you got your green-red machines. And all that's really going on there is we're making different versions of the machine, but they're in a living system, a cell. But this particular machine is a DNA polymerase. It knows how to remake DNA. And so what we do is we say, DNA polymerase, we will now put you in a water and oil emulsion. And again, the words wash over you, and you're like, what is a water and oil emulsion? Okay, next time you have salad, And you're sitting there going, I think I should like some oil and water for my salad. And you have oil and water, and you go shakety, 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 and it turns into this sort of misty mixture. That's an emulsion. All that's really happening is all the water has dispersed throughout the oil in tiny, tiny little bubbles. So in tiny, tiny little bubbles, we have tiny, tiny little cells. But now we blow the cells up. They're dead. They are truly dead. They are not coming back. But inside were the machines that know how to make DNA. And so we just asked them in their little water capsules to make more DNA. And they do. And some of them that are green make green, and some of them are red make red, and if red is better than green, you make more red than green, and then you go through and make more. This is evolution in a nutshell. You have genetic diversity that is heritable, first via a living thing, and then via a non-living thing. And if that diversity makes more of either the living thing or the non-living thing, the polymerase, then it can survive and multiply. And so in this regard, we use the cell as a big bag to carry around our machines, but then we dispense with the bag, and we just use the machines directly. And what Jared Ellington and actually this concept, I thought about it this morning. How do I get this concept of you don't really need to be alive to be useful across to y'all? And I thought of two examples. It's not really as weird as it seems. We transfer lots of things between us. We benefit from dead people all the time in the form of transplants. Your driver's license probably has something that says you will or won't help other people once you are dead. And similarly, we have even non-biological feed forwards, inheritances, and you can think of lots of other ones. Social networks, all sorts of different ways that we feed forward even after we're no longer here to bedevil the universe. And so similarly, it's not, with that in mind, it's not too ridiculous that my bacteria have little engines that feed forward on systems even after they're dead. And so what we did was we said, hey, what if we now make you replicate through RNA rather than DNA? We set you this task. And again, you start out the task by just saying, I get made in a cell, but then the cell gets blown up, and all I'm going to do is see if I can replicate RNA. And to make a long story short, Jared Ellington in my lab worked really hard at this. And over many generations of directed evolution accumulated a new type of polymerase that can do reverse transcription, can do this process that normally only viruses do, and do it very, very well, and do it as a DNA, from the lineage of a DNA polymerase rather than an RNA polymerase. And this is useful. We can use this for diagnostics. This is actually us using our new polymerase to amplify particular genes from mixtures. We're actually beginning to use this in assays to look at things like Zika virus, because as you might imagine, it's sometimes difficult to amplify small amounts of information, small amounts of sequence information to tell if something is there. This is a great enzyme for that. And actually, we found out that even though we evolved it for RNA, it can work on weirder things. That's not RNA, that's a variation of RNA. And so we're looking forward to making a whole variety of so-called genetic alphabets. Our genetic alphabet, DNA, RNA, GATC, a little too limiting. So we can bring in things like new types, new chemicals, and potentially expand the genetic alphabet, thus expanding the tree of life. So change the machinery, change the game, change the tree. Next, we decided to try this with an RNA polymerase rather than a DNA polymerase. So rather than reading RNA into DNA, we're now going to read DNA into RNA slightly differently. This is again work by Jared. There's Jared being happy because he's almost ready to leave my lab. And what we did was we created new RNA polymerases. And actually, I'm belaboring the point here. I really am. Oh, good. I didn't belabor it too much. I couldn't remember how many slides I took out this morning. So as the great sea of information washes over you and you're like, I don't understand, just take it from me that we can change this RNA polymerase. And now it's going to essentially use a different sequence to start transcription. And this is now going to lead us to be able to make new organisms, bacterial viruses, but still organisms, that get used to their new parts. And this is the point, right? Previously I said in a top-down way, from large to small, we have a goal for the bees, and we back-engineer to meet our goal. Here, we're going from small to large. We have new machines. We've evolved the new machines. The new machines give us the possibility of developing new branches on the tree of life, but what will those branches be like? This is more of a discovery effort. What do we get when you change the code? What do you get when you change the basic chemistry? And so bacteriophage are really good because they're tiny, they replicate really quickly, they're easy to analyze. And this is Dan Gary, who went through and took one of our new RNA polymerases that, again, all the letters here don't matter so much. It just recognizes a new promoter, recognizes something in the bacteria, and then we force the bacteria to get used to it, right? Previously, we're going to addict the bees, right? We're going to say, you must be used to this. Here, we're going to say, if we give you the option of using this, will you like it or not? So we're being a little more engaging with the organism. But it's a really interesting question. If I gave you new chemistry, could you use it? If I gave you a superpower, could you use it? That's actually a bigger question than you might think, right? Because it's like, what if suddenly I could just, fly in the sky? I don't know. I'd probably leap, really high and be crushed by an airplane. I don't know. I wouldn't know necessarily how to use my new superpower. So if you've given an organism a new chemistry, a new superpower, what does it do? How does it adapt? So we teach it to adapt by just saying, we'll give you a long time to grow, many, many, many cycles. It's like we have zebra on the veldt and they get killed and one zebra goes and, or two zebra goes and starts the new family and it grows and then they get killed by the lions and then two zebra goes and starts the family and so forth. So that is just the same. It's just bacteria and bacteriophage. And what happens is, here's the parent and it takes it a whole long time to grow its way out of the cell. And you're like, wait, that's going down. Yes, it's growing its way out of the cell by killing the cell. All right, it's sort of like alien popping out of the cell. Out of you, it's sort of like you would go down. So the bacteriophage popping out of the cell kills the cell, but that's okay. The bacteriophage is fine. And over time, it gets faster in the presence of its new, fun machine that does different things. Now, at the level of actually doing science, we scientists have to, piece things down to individual little parts. And so I tell you this bacteriophage has a new machine, and I circle in red the places where we think mutations might occur to accommodate this machine. And in fact, we find many such mutations, right? You do not have to apprehend this. I don't think I apprehend this. It's just to say when we looked for places where the mutation should arise, they did, and they sort of make sense. In terms of what we expected this to mutate to, we expected it to mutate from this to this, and it sort of did, but it also found new things. And this is what's so cool about evolution, is that you give it a chance in the test tube to show its stuff, and it does sort of what you want it to do, but it also comes up with new ways. It's the old Jurassic Park, life will find a way. It really does. And so I gave my little bacteriophage rather than big dinos. I would much rather be working on big dinos, incidentally. The way to evolve through a test tube, and they did, and they picked paths that I thought they should, and they picked some new paths I didn't think they should. And this is just Dan saying that this thing has now learned to accept its new machine, but it actually also still likes the old one. This is another aspect of directed evolution, is that while I fervently wish that life would choose my new chemistry, that it would choose the new superpower, it's really pretty reluctant to. And maybe another way to say that, if you had your new superpower, and you could jump really high in the sky, but your legs had to be 12 feet tall. Would you want to have 12 feet tall legs if you could jump really high in the sky? I don't know, maybe. But it's like, I'm having to make a trade-off. I wish there were some other 12 feet tall people, or maybe I wish I could shrink my legs up on occasion. Again, there's lots of trade-offs, and getting the new form, the new chemistry, the new way to work is not always easy. Now here's a second experiment. And it's again with bacteriophage, and was carried out by my colleague Jeff Barrick, the guy that wants to slow down evolution, except not this time. This time Jeff wants evolution to occur, and he's feeding the bacteria an unnatural amino acid. So we're back to the ye old genetic code. We're back to changing the underlying machinery to change the genetic code. But we're going to put in this new amino acid. Previously I talked about nitrotyrosine. This thing's called iodotyrosine. Great, it's another chemical. And Jeff found that, again, when he did serial passage, serial passage, serial passage, zebra lion, zebra lion, zebra lion, zebra lion. Things grow, they get eaten, they grow, they get eaten. They found a bunch of mutations, right? Just like before. But this was really cool. He found 3 types of mutations. First, he found things that just sort of tried to repair themselves. They didn't like the unnatural amino acid and what it was doing, and so they figured out ways to repair it. Yeah, you might expect that. He found things that were neutral. It's like, sometimes I'll adopt the unnatural amino acid, sometimes I'll adopt the new chemistry because it just doesn't make any difference. You can't tell my legs are any longer. It's not really a superpower. It's okay. But in one case, he found it actually pushed forward towards grabbing the new chemistry, the new amino acid, adopted it And it was because it helped the phage break out of cells. It helped the phage lyse the cells. And he actually proved when he did competitions between non-new chemistry and new chemistry that new chemistry won. Okay, so this is actually, and this is pretty significant. I think this is the first known example of positive selection for the adoption of new chemistry. And Jeff doesn't always tout it that way, but the more I think about it, the more I think this was really a watershed moment in biology where an organism wholeheartedly adopted a new chemistry. It said, yeah, give me that superpower. So that's sort of interesting. All right, now, that was, again, at the outset I claimed, A, amorality, B, that information would wash over you, and C, that I was lazy. And I think I have proven all of those. But I hope that it has been modestly interesting to you, if only to figure out something about, step back from the information, where science can go or could go, where technology can go or could go. And again, you constrain me. You are my masters. And so you don't have to really go, oh my God, we must do something to shut this all down. You as a populace, as an informed populace, get to choose what people do or don't do. I'm a technologist. I make technologies. They do stuff. You decide where it all goes. But I'm pretty excited about, one, being able to do top-down engineering to create new types of living systems that can potentially do beneficial things, and bottom-up systems where I can create new ways to engineer the tree of life, where the tree of life decides where it's going to go. And that's exciting to me too, because already we're finding sometimes we want the superpower, sometimes we don't. Sometimes things change in ways we know, sometimes they change in new ways. So I thank all the researchers who contributed to this research. I showed you pictures of some of them all along the way. All the sponsors, the people that give us money, really important. Money, very important. You for listening to me, because an engaged populace is the safest way to make sure that whatever form of morality you favor comes to the fore. And again, knowledge is a good thing. But finally, given the day, I thank Big Chuck and for his inspiration, for his starting it all off, as it were. I think, whether he would recognize the fruits of his labors at this point is unclear. The fact that we can now, at a molecular scale, do evolution and direct the existence of organisms, I don't think was necessarily something that Charles Darwin thought about. However, if you read his grandfather Erasmus, I think Erasmus pretty much had it hacked. Erasmus was a smart guy. He didn't have quite the tools Charles did, but he definitely saw where things could go. In any event, thank you very much. I hope I haven't wasted too much of your hour.